Yo, what's up, guys? So, welcome back to B-iTalk podcast, where we discuss biomedicines and bioinformatics in a nutshell. So I'm Albert, your host for today delivery, which is the fourth delivery, and we are joined with our hosts Enrique and Karin. So you might have heard their voices before the podcast as speakers for biomedicines and cancer immunotherapy. So let's welcome them once more. Hello, guys. Say hello to the viewers.
Hi everyone, I'm Karin. I'm previously with you guys in conference on the cancer immunotherapy session. Nice to meet you all again.
Hello guys, my name is Enrica and I'll be your third host for this session. We will be talking about computers in science. I'm referring to bioinformatics and we'll be covering several BI topics such as sequencing and NGS, along with several of its application in life sciences. I highly recommend you guys to listen to our first episode podcast to understand bioinformatics in a nutshell. That is linked in the description. And come back to listen to this episode.
Without further delay, let's introduce our speaker for this topic, who is also the only bioinformatics student in our team, and our chillest member, Jeremy.
Hi everyone, I'll be your speaker for this session and I'll be explaining several bits of what bioinformaticians do.
Okay, so let's just start it, shall we? So in the first session, you mentioned that the bioinformatics is a relatively new field and it uses computers to analyze biological data into useful information. So I'm curious though, how different is this field to regular computer science? Is there a lot of coding as well as programming?
The majority of tools that bioinformaticians use are in the form of programming languages or scripting in the terminal. There is Python or R language that is frequently used, while for the scripting it is done in the Linux environment, which is also very similar to the Mac operating system.
Okay. Hold on a bit. Yeah. So, most of us, probably like me, does not understand one of those familiar with programming. So, let's step back a bit. So, please explain what is Python and the R programming language, so we all on the same pages.
Both of them are programming languages that computer science students study. They can be used to create programs and also perform analysis of data. One of them is Python. It is a general purpose programming language that can be used to create analysis of any kind. R, on the other hand, is a more specific programming language. It is mainly used for statistics and analytical processes, but not many programs are made using R.
Wow, you need to study both computer science and biology? Seems like a really difficult field of study.
Yeah. So, okay. Let's dive into something more specific, like that combines both of the science and the computer science and the biology part. So, we have talked about the involvement of BM with the vaccines and the PCR during the pandemic, since the pandemic is a major issue that is affecting all of us. So, are there any involvement of bioinformatics during this COVID-19 pandemic?
Okay, sure. There is the sequencing technology that has been developed to become very affordable. It's called the next generation sequencing, or for short NGS. It has helped scientists identify the various strains of the SARS-CoV-2 virus, such as the Alpha, Delta, and Omicron strains from the genetic makeup of the virus.
Cool! If I'm not mistaken, the difference between the strains lies in the mutation. What does this refer to?
Well, the viral cause of COVID-19 pandemic is called the SARS-CoV-2 virus, and this virus belongs to the same family as the SARS virus that caused the SARS outbreak in 2002 and 2004. This means that the genetic material of the virus, in the form of RNA, has a lot of similarities. The mutations that happen in this virus lies in the RNA, and most of them are small changes in the sequence.
Okay. So I have a question though. So when you talk about small changes, how small are you talking about here, and what does sequencing technology have to do with it?
Mutations can cause single nucleotide changes called single nucleotide polymorphisms, or SNPs for short. However, there are larger mutations that change more than several nucleotides in the sequence. Mutations that affect the whole chromosomes are generally very rare. In simple terms, sequencing technology reads the genetic sequence of a sample, which can be in the form of DNA that humans have or RNA that the virus possesses. Once this sequence is obtained, an identification of who that sequence belongs to, the differences in nucleotide variations, which is to know the presence of mutation, creating a phylogenetic tree to know the familial relations, and so much more is available to do for bioinformaticians.
I see. So, you have mentioned about the possibilities that sequencing can do and also its contribution during the COVID-19 pandemic. Other than this, what are other applications of sequencing technology in life science?
Well, the applications are actually limitless. It can be for screening inherited diseases, human genetic studies, reproductive health diagnosis, microbial and cancer research is also possible. It is also used in gene editing and also drug discovery efforts in the medical field.
Interesting. Okay. And we would like to say thank you to Jeremy for the delightful insights of this podcast. And if I'm not mistaken, for our next session, we'll be covering drug discovery, right?
Okay, hold on. Hold your horses, gentlemen and girls. Let's get the details for the next session. Yeah, we'll be talking about the collaboration between the BI and the BM.
So that's it, guys. We have come to the end of our session today. For those of you who want to know more about BI and BM, stay tuned for our next section. Bye.
Bye. Take care then. Bye now.